and welcome. VRL is excited to share details about our latest offering, adeno-associated virus screening. If you are not familiar with VRL, you would appreciate that we have worked the past 30 years in animal health monitoring and diagnostic testing, with a global presence of 2 facilities in China, just outside of Shanghai, and 2 in the U.S., San Antonio, Texas, and Gaithersburg, Maryland. VRL screens at least 60% of the global NHPs. The VRL technical team, under the direction of Dr. Qiling Zhao and Dr. Arlene Leon, worked long and hard to provide a more efficient and productive testing methodology, setting a new standard for the advancement of gene therapies. I will now take you through the history of this program's developments, formats, and performance characteristics. Gene therapy has become one of the most promising therapeutics in the treatment and prevention of diseases. Gene therapies uses modified viruses or other technologies to deliver therapeutic genes to address genetic diseases at their source. Among the technologies available for gene therapy, the adeno-associated vector, or AAV, technology has become the most used, as it effectively delivers therapeutic genetic material into cells or tissues. Once the virus enters the cell, the therapeutic gene will move into the nucleus and the gene is expressed. The AAV program, developed by the VRL team, is a screening methodology that was designed to identify AAV antibodies in serum samples. The VRL neutralization antibody assay test depends on the ability of viral particles in the assay to infect cells containing a GFP reporter. In serum that does not contain AAV antibodies, the test viral particles will infect the cells and turn on the GFP reporter, and the signal is detected. In serum where AAV antibodies are present, the antibodies will bind the test viral particles and inhibit its ability to infect the cells. In this case, the GFP reporter will no longer produce a signal. The AAV NAb test will detect antibodies against several AAV serotypes, including 1, 2, 3, 5, 6, 8, 9, and 10. The VRL AAV program offers 2 levels of antibody testing. The first one, the NAb screening assay, helps facilitate screening of research animals to be used in a study by identifying animals that are negative for the AAV serotype. Those animals are often used for the development of gene therapies. The screening assay is a qualitative neutralization assay. The result is reported as positive or negative for the 3 dilutions: a 1 to 10, 1 to 20, 1 to 40 of each serum sample. The second test, the NAb titer assay, is used to identify the presence of the transfected AAV particles containing the therapeutic gene. The titer assay is quantitative neutralization assay. Each serum sample is 2-fold serial diluted from 1 to 10 to 1 to 5,120. And the result is reported as a titer value at which 50% inhibition of virus transduction is calculated. The quality performance of the VRL AAV test has shown that the assay has a high analytical sensitivity and specificity. As of today, the analytical sensitivity and specificity have been determined for several serotypes. The VRL AAV program provides the convenience of results in days instead of weeks, high throughput, competitive pricing, and all with the quality diagnostic testing you expect from VRL. We are very proud to be one of the first and only laboratories offering a commercial AAV testing program. This is yet another example of VRL's commitment to innovation and research diagnostics.